Hi, my name is Eric Sokol, and I'm a research scientist with the National Ecological Observatory Network. Today, I'm going to give an overview of NEON's terrestrial observation system. NEON's data collection methods include automated instruments, observational sampling, and airborne remote sensing. Today, I'm going to focus on observational sampling, where essentially humans are the instruments that collect samples and field observations. NEON's terrestrial observation system produces 40 data products. These inform 3 data themes, including organisms, populations, and communities, land use, land cover, and landscape scale processes, and biogeochemical pools and cycling. TOS sampling targets a number of sentinel taxonomic groups to provide data to characterize diversity, abundance, pathogens, phenology, biogeochemical pools and fluxes, metabolism, and productivity and biomass. Additionally, we collect soils to provide data on biogeochemical pools and fluxes. The letters and symbols above the column headers on this table indicate which sorts of samples are archived and how. The observatory has 81 field sites, 47 of which are terrestrial, and produces 181 data products, 40 of which are from the terrestrial observation system. NEON's terrestrial field site selection is based on a number of criteria, including a minimum of 1 site per domain in an area that is considered managed as wildland. Additional sites may be added in a domain based on other interests, such as detecting invasive species or changes in land use during the life of the observatory. Lastly, terrestrial sites are co-located with aquatic sites where possible. Okay, so now I'm going to give an overview of a terrestrial field site. And you can find more data about spatial and temporal sampling strategy in the document listed at the bottom of this slide. The figure on the right is an illustration of an idealized NEON terrestrial field site. It doesn't represent any one particular NEON field site, but gives us an idea of all the different components of a NEON field site. The legend illustrates the different types of plots and transects that occur at a NEON terrestrial field site. And I want to point out 2 different scales of sampling that the observational team uses. So first we have site scale sampling that has up to 30 distributed base plots, and these are spatially balanced and stratified random. And plots are allocated in proportion to NLCD cover type. Second, we have airshed scale sampling, where we have 20 to 30 smaller plots all located within the airshed of the instrument tower. Here we have spatially balanced randomized sampling, but it's not stratified by NLCD class. And so data coming from these plots is meant to be linked to data coming off the instrument tower. Now I'll give a little more detail on distributed base plots. So again, distributed base plots are meant to characterize the site, and there can be up to 30 distributed base plots at a site. Many different types of observations are co-located within a distributed base plot. On the left, we have a schematic of the spatial design of a distributed base plot. So these are 40 by 40 meters. And then there's a central 20 by 20 meter core plot. And within that, there's a number of subplots that are nested. So you'll need to look at the documentation for a given data product to get more details about how those data were collected within the context of this plot. On the right, we have listed a number of different types of data that are collected within these distributed base plots. And I'll point out that plant diversity is implemented at all the distributed base plots at a site. In some cases, it's been scaled back a little bit from 30 to fewer plots if our optimization analysis justifies that. We also collect plant and soil coordinated sampling from a subset of distributed base plots. So often up to 10 plots, we do that. And then ground beetles are also collected at distributed base plots. There are a number of sampling protocols that have their own spatial sampling design, but they're coordinated with distributed base plots. So for example, for small mammal sampling, small mammal trapping grids are located adjacent to a subset of distributed base plots. So we have an illustration of the layout of a trapping grid in the center of this slide. The small mammal sampling is meant to provide data to characterize populations and communities and also individual traits. And samples are collected for blood analysis and sent off for blood pathogen testing. There are a number of other data products that have their own spatial sampling designs, again, where they're either traps or transects or plots that are specific to that protocol are co-located with distributed base plots. So ticks and mosquitoes are examples of other data products where this is the case. And Sarah's going to talk more about those later today. Lastly, I'll talk about phenology. So this protocol is meant to characterize phenology of the dominant NLCD class for a site. And phenology plots are located within the instrument tower airshed. And here we have two kinds of plots. So there's a phenocam plot where the phenocam captures images every 15 minutes. And then there's a primary phenology loop where field scientists walk the loop and collect phenology observations up to 50 times a year. And so this data product, for an observational data product, is one of the higher frequency data products and may be better for ecological forecasting. And I know that some folks have already been conducting forecasting studies using these data products. Okay, now I'll talk about temporal sampling design. So first we have coordination across years. This table is an example of coordination for plant and soil data sampling. And so, for example, we have below ground biomass in the first row of this table, and it has an interval of every 5 years. And it's sampled at anywhere from 20 to 30 tower plots. And if we look a few rows down, we have DIV, so that's plant diversity, and that's sampled every year at distributed plots. And some of the plot numbers here may be scaled back based off of our optimization analysis. I want to point out that for the every 5 year sampling protocols, they're somewhat staggered so that we don't have a spike in labor all in 1 year. We also have some temporal coordination within a sampling season. So there are multiple bouts per year at regular intervals or at seasonally important dates, or a single bout per year timed to a seasonally important date. And so those data products are listed here. And the timing is queued off of the MODIS Enhanced Vegetation Index. Lastly, I want to mention that we have our observational system data pipeline, and you can learn more about that in the YouTube link below. We have a schematic of it on the right here. And also you want to consider data latency. So there's 4 types of data that go into our pipeline. Field observations, samples analyzed in NEON lab facilities, samples shipped to contracted facilities on a rolling basis, and samples shipped to contracted facilities on an annual basis. And latency can vary from 2 weeks to 2 years, depending on the workflow for the data set. And so thank you for listening, and I'm happy to take questions during the course of this meeting.